Perfect. So welcome everyone. We are so glad you're joining us today. My name is Michael and I will be moderating today's session, which marks an exciting milestone for Q Biotech, the launch of our NativeMP Plate set. Before we dive in, a couple of quick housekeeping notes. So feel free to drop any questions in the chat. So you have a chat on the right, just type it in there. At the end, we will collect them and then Philip and I will answer them. You will also notice a poll running on your screen. We will ask you how many hours per week do you spend on your manual pipetting. We would love to, if you can, during the webinar, just leave your poll there. So it should run now.
Yes.
And now on to the good stuff. So we all know how challenging membrane proteins can be to work with. And yet they represent one of the most important target class in drug discovery. Around 60% of all approved drugs target membrane proteins, but fewer than 2% have been structurally characterized. This gap is exactly what we are here to address. The NativeMP Plates set gives you a straightforward, scalable way to stabilize membrane proteins in a native lipid environment in a plate format that fits right into your existing workflow, without the drawbacks of conventional detergent-based methods. And now speaking of the workflow, so we have, as I also announced on LinkedIn, we have something special for you today. So we have recorded, yesterday, the complete NativeMP workflow running live on the Kingfisher. And we are going to start it right now. So we will now, during the webinar, watch there from time to time going in there. So now Philip is now pressing play. So it's now starting. And we will, during the webinar, we will look through it and how it went. Also that you basically can see, okay, how fast it is done, and we'll just take this hour of the webinar, then it's done. And yeah, perfect. So thank you, Philip, for putting it up. Now it brings me to our speaker that is here. So it's Philip Harnisch, someone who knows the workflow better than anyone else. He is head of the lab at Q Biotech, and Philip has dedicated his career to membrane protein research. He started with detergents in the lab and has been through all of the struggle, all of the tears and hard work.
Multiple times.
And then when he joined us, he learned about the way of copolymers, which at the beginning also was not easy, but then through time, he brought the technology forward with his team, and now we are sitting here. So he completed his PhD at University of Düsseldorf and used it extensively. So I think Philip, we take it now from here. We're looking forward to your presentation.
Yeah.
Thank you very much.
Thank you very much, Michael. Yes. So what you're going to see, or what you're seeing right now here, is already solubilizing membrane proteins. And as Michael kindly introduced, we're going to go back and forth. We're going to have a presentation. We're going to lead you through the workflow, how it's working, so you understand what you're actually looking at here. And then, from time to time, we're going to check how far the robot is. And therefore, you're going to see that within the hour where we are going through the presentation, that we will see that membrane proteins are getting purified nicely. Okay, then let's dive in. Okay, so a little bit of introduction. So why are membrane proteins still a bottleneck, or why are membrane proteins important at all in the very first place? There are so many proteins out there. Why did we at Cube and many of you and many people in drug discovery focus on membrane proteins? So on the right side, you can see the 2025 Q2 top-selling drugs from the whole market with market capitalization. And every membrane protein, every drug which is in green has a membrane protein as a target. So it's obviously the majority, even though the membrane proteome only makes a small number of all the genes in the human cell. So we can say that 60 to 75 of all top-selling drugs are actually targeting membrane proteins. And over many, many, many years, including my studies and my PhD, detergents were the gold standard. Everybody was using detergents. Literally nobody, except a few little rebels, were using copolymers in the beginning. But we all knew from the very beginning on that detergents come with a lot of trade-offs. And one of the trade-offs is they come with low stability. Once the membrane protein is out of its native structure, A clever man once said, if you put an iceberg to the desert, it's not very happy. I like this very much. And I also like to say, like, I'm more happy in my living room where I have my sofa and my television than outside when it's raining. So it's a little bit like with detergents there. It works somehow. It sometimes works very good, but it doesn't work for everything at all. Plus, this comes with like high hands-on time, low reproducibility, and it definitely requires expert. So it limits the scale in pharma and in academia. Okay, so what we do at Cube here is we do not work with detergents. We do work with copolymers, and copolymers are amazing because they're very convenient. You only need to add them in one step in the solubilization phase. They find their way inside of the membrane and they cut out so-called native nanodiscs, as you can see on this slide here. So they're going to wiggle their way into the membrane. They're going to cut out those nanodiscs. The copolymer belt stabilizes the membrane protein in its native lipid environment. So we can retain the lipid environment the protein likes best. It can stay in its living room. It can keep its television, and it can be happy. And actually, this works wondrous. You're going to see how good it works. And therefore, we can retain function and stability. We can purify at room temperature. We can perform assays at body temperature. We therefore have lower risk and also faster timelines. And what we are very proud of, we are now able to work with full-length wild-type GPCRs. So no modifications whatsoever, except one tag to affinity purify over overexpressed membrane protein. But we don't have to do any stabilizing mutants because we have the copolymer, we can keep the lipids. Okay, so over the years, there have been many copolymer backbones introduced, and we have covered them all. You can get them from our website, 32 different copolymers from 5 backbones. And there are some older backbones where the whole community started with, like SMA and DIPMA. Then in 2021, 2 new backbones were introduced, the ultrasoluble amphipoles and the ASTIs. And in 2025, we came up with our own copolymer backbone, the Cubipol. So all of these copolymers, they work more or less the same, so they can self-introduce themselves into the membrane. They all cut off native nanodiscs, but they have different site modifications. And of course, the backbone chemistry differs between the different copolymer backbones. And what we have found out, and also what the community sees, there's no one fit for all. So you have to do screening, and we love to say screening is key, because it literally is. You have to screen for the perfect copolymer for your specific target. Okay, but there's many, 32. It's much. And if you have to do it by hand, it's also very much work. Additionally, we are able to secondary modify the copolymer belt itself. So we can add the biotenuity, we can add the fluorescein. We have this also directly available from our website, or we can introduce any custom modification any customer wants. So if you're interested in a specific modification, you already found your copolymer, you can come to us and we can make anything possible, basically. Okay, so 32 copolymers is much. Plus, there are bottlenecks in membrane protein work. Like with detergent, it takes a lot of time. And I would like to introduce you now to the classical workflow people used to do with copolymers. And also we, 5 years ago, were doing it exactly like this. So we had to get our cell pellet, we had to resuspend it, and we had to lyse it. Then we were clarifying it, getting rid of the cell debris, creating a so-called clarification step, 9K supernatant. With this, we then went into the ultracentrifuge for at least 1 hour. Some people even do it for 3. We were pelleting membranes. Then this membrane, we were adding a copolymer stock solution to, and this then incubated overnight to solubilize as much as possible. The next morning, you come back to the lab, you spin it again, you make a clarification post-solubilization, again ultracentrifuge. Then, with this, you go into your resin, you bind it, you wash it, better more than less to get a pure result, and you elute. So, there's many steps, and we did everything by hand. So, my question for you is, like, how many purifications can you actually perform in parallel when you're working in the lab? I know it for me is like 32, but it comes with bleeding thumbs. It comes with that I'm annoyed and I don't like this so much. So we were thinking about how we can actually save this precious time, because it's 20 hours from pellet to eluate. And then you have not seen a blot, you have not seen a gel, you have no idea if your protein is actually there or if you just worked with some nice buffers all day long. So there's no quality assurance, and you have no idea what you actually have. And it's still high hands-on time, and it's very hard to reproduce what you did on a Monday the next Tuesday. So we still require experts. There's still a limited scale. Plus, our customers, they come to us, we talk with them in online interviews, we talk with them on conferences, and they tell us all the time, I don't want to screen all those polymers by hand. It's too much work for me. Just give me 1 or 2, and then I do it, but I just stick with detergents. Because it takes too long. How can I be sure that my protein is best quality? I have huge variance in my repetitions, and I have literally no experience at all. And sometimes I'm also afraid that it does not work out. So I've established a detergent, it sometimes doesn't work, but then I just modify my protein. I don't want to hassle with this. And this stuck with us. Because it's true, like it's so many steps and it's so much work. Like you have to label all those tubes. You have to weigh every sample. You have to prepare those copolymer stocks. I have to calculate 100 times. I have to pipette everything, label again. I have to mix it, balance the tubes. I have to do all these centrifugation steps. I have to wait really a lot. And this is often times where I cannot do anything else. So I'm just standing in the lab and I'm wondering what to do with my life. Then you transfer all the supernatant, you set up all your purifications. If you do it manually in like drip columns, it takes forever. It's annoying. If you do Mac beats, it's expensive. If you can do it, it's great. Then you have to document everything by hand and you analyze everything. You do gels, you do this. It takes a week to just get one screen done. Yeah, so it's annoying. So we thought, okay, what can we do? And we figured out we can actually shorten a lot of steps. We can shorten the lyses. Okay, saves us 7 minutes, better than nothing, but not much to talk about. But the cool thing is, like, we can work from 9K supernatants. So we don't have to do all this ultracentrifugation. We can actually cancel ultracentrifugation from our screening completely. Like, this saves us a lot of time. It saves us a lot of resources. It saves electricity. That's also great. And we can work from this. Like, if we are using the automation technology in Kingfisher, which we are showing you today, you can bind it for 5 minutes and solubilize it at the same time. Yeah, that's great. It can even save you. Or you do a little step, 5 minutes solubilization. If you're a little bit afraid to do it 20 minutes, it's also fine. Saves you much time. It goes so quick. You bind it to the resin, you wash it. Washing takes actually longer than binding it to the resin, and you elute it for 5 minutes. And then you take your eluate and you spin it down to make sure there's no aggregation or some residue of Mac beads in there. And then you're done. And it's so fast. It's like 1 hour. I mean, we can have a quick look. The Kingfisher is already collecting Mac beads at the moment. So, or is it stuck?
He's collecting.
It's collecting. Yes, it's collecting Mac beads already. So that's great. So how do we do it? Of course, we have to make it convenient, so you don't have to worry so much. So we developed 2 plates. One plate has copolymer inside. It's our NativeMP screening plate. And one plate has Mac beads inside. That's the purification plate. And we can add them to the Kingfisher conveniently. And we developed a protocol. So you load it onto the Kingfisher, you press play, it tells you where to put which plate, and then you press start and it goes on. Yes. So then you can start to worry, like, which copolymer is actually the right one for me, which stabilizes efficiently, which gives me the best output. And yeah, you can now save time on purification. And you can actually work on assay development. Okay, so how does this look? So for now, everything is made for Kingfisher Flex, but we are onboarding it to the newest model, Kingfisher Apex. So this is coming very soon. So the plate type are those specific Kingfisher plates. You're going to need those plates because you have this tip comp. It has very specific layout. And yes, we bring everything ready in those plates. Okay, next question is, which tag do you have? So we love to use the Rho tag, Rho1D4 tag. We think it's an amazing tag for membrane protein scientists, and we use it in-house all the time. So this is an option. Okay, you're using with working with strep tag. I can totally understand this. We brought strep tag Team XT on there. Or also for GPCRs, very often people use flag tag. Okay, so we got those Mac beads also there. We put them on the plate for you. Next thing is, you have a pre-selected copolymer screening. So we figured out what are the 16 most prominent or the best copolymers for you to get success. And we have them in there. So we can have 3 different sets, basically. You have 2 screens in one plate, you have 4 screens in one plate, or you have 6 screens in one plate, and you can just add to the basket the fitting purification NativeMP plates to complete the set. Yeah, this is how it looks. Okay, so these are the 16 copolymers we figured out. Of course, there's our Cubipol series in there because it is the only copolymer backbone which includes all site modifications which are else only available on other backgrounds. There's the ultrasolid amphipoles in there, 2 ASTIs, the classical old gold standard SMA 200, 2 sulfur variants, and 1 DIPMA variant. You have 25 milligram of copolymer per well, and you just have to add 1 milliliter of your lysate. So 1 pellet of 300 milliliter expression culture, overexpression culture, divided over 16 wells is enough for 1 full screen. So basically in 1 hour, you can get 2 full screens done if you use this plate. Okay, for the MAC beads, it looks like this. We have 30 microliter of MAC beads per well. And what we are very proud of is that those MAC beads are specifically formatted. They are dehydrated. So they are little platelets. I'm going to show you later. And they're very easy to store. We can ship them all around the world. Easy peasy. There's no spilling. There's nothing. This is our patented film technology. And this is basically how it looks. So 3 different sets. 2 screens, 4 screens, 6 screens with the corresponding MAC bead plate. You can see the copolymer finds this gets this little lyophilized cone, and the MAC beads films, they look like this. Okay, now you're saying maybe, okay, this is not enough copolymers, I want more, or this is not the right copolymers for me, I want others. No worries, we get you covered. With a minimum of 5 plates per order, we did a custom for you. You tell us which copolymer would you like, how do you want the magnetic bead types, so we're open for suggestions. How do you prepare your cells? So I showed you the little graph, how to prepare them, but maybe this is a bit more visual. So we have an overexpression membrane protein culture of 300 milliliter. This is how we do it in-house. You have more, it's fine. You have less, you have a good expression, also fine. You verify that you actually have overexpressed the membrane protein with a specific tag. That's important. If you have nothing inside, you cannot purify anything. You spin down the cells to a pellet. You add buffer, e.g. 2:1 ratio, you open the cells, we'll have to do sonication. You do it differently, no worries, you can do it. Just make sure that there's no DNA inside, maybe add DNAs, else it's going to be a sticky thing afterwards. You just spin down your cell debris for 30 minutes, and then you check the nanodrop, how much protein overall you have in. This is an indicator for us that we can make sure that the solution doesn't get too viscous when we add the copolymer later, and then you're ready, set to go. And then you have 2 simple steps which you have to perform. First, you have to equilibrate your magnetic bead plates. You can do this while you have the centrifuge running with your lysate, so it doesn't kill you any time. You just add 1 milliliter of wash buffer to the MAC beads wells, which are filled. You add 3 deep well plates with washing buffer and a tip comb into the machine, and then you start the plate equilibration protocol. We send you over, takes 20 minutes. Kingfisher is going to equilibrate the beads, wash the beads, and leave the beads in the 3rd plate. And then in the next step, you can, in part 2, you can use this very easily as your MAC bead plate. Everything is prepared inside. You just add your lysate to the copolymer plate. You add the TIPCOM plate, 3 new wash plates, and 1 elution plate, and you start. You press play, and it starts purifying. And with the full plate, you can have 96 purified membrane protein samples in just 1 hour. Very convenient. So to sum this all up, pipette, buffer, and lysate in plates, press the button, wait a little bit, and 6 screens with 16 copolymers each, and 90% time saver. So actually, it's more work to pipette everything out of the plate later than to actually perform the experiment. Yes. Okay. So how does such a screen look? You see here the screening plate, purification plate. This is exactly how we did it. We did everything on Kingfisher with exactly the protocol we are providing you. This is our little in-house protein we love to use. It's P2X4. It's a trimeric pain receptor, also an ion channel, has a big ectodomain, and it was a very hard target back in the days. So in detergent, it took 20 years to get a structure. Literally, it was published last year. In copolymer, it looks like this from 1 pellet. So you can see there's difference in the different copolymer backbones. Here, the next generation copolymers, as the ultrasolute copipools, they perform very well. Some SMAs perform very well. DIPMAs, in this case, didn't perform so well. We also made a gel analysis on the right, where you can also see this in a box plot. Okay. We also did this with many, many different targets. And these are, how many targets are this? So 13 targets. So it's a little bit more than 2 plates. So you could literally do all of this work in one day, which back in the day would have taken us many scientists, many hands, and many weeks. And there's GPCRs in there, RDRB2. There's the amylin receptor calc-RAMP, which is a super high-class target. We have claudins, we have HERG. It's a huge protein. We have SLCs, which are notoriously hard to get. We have CD proteins and also an EGFR. So you can see small proteins, large proteins, huge proteins. Everything is possible in the same setup. All you need is an expression, a lysate, you pipette it in. But what you also see is that there's difference in efficiency. So we just pasted under the best performing copolymer for this specific target. And you can see sometimes it's ultrasolute 80, and sometimes it's an ASTI, sometimes it's an cubipole, and also sometimes it's a DIPMA, in this case glycodipma for EGFR. And if you look at all the targets, and we just analyze all the protein we got out of there over the 13 targets, you can see that in general, the next generation copolymers, they perform significantly better than the old school backbones, DIPMA SMA, and in this case also the SMA BZs, in output. Okay, of course, this doesn't mean that the protein is perfectly suited for your specific analysis. It could also be aggregated. We don't see this in this case, but bear with me. Later, I'm going to show you. But first, what does this mean for you actually? But what's the scientific impact of using such an automation? Okay, of course, you can explore larger experimental spaces, the same amount of protein. We need very little protein. You can evaluate the copolymer chemistry, especially temperature and buffer conditions simultaneously. You're going to see in the next slide why I emphasize temperature so much, because I can show you something which shocked me literally. You can rapidly identify the optimal membrane protein stabilization conditions. You can, of course, generate way more data per experiment. You can definitely improve reproducibility between the users, the project, and between laboratories. We have done some experiments in labs in the US with the same output as here in Monheim with us, which is already worth mentioning because this is definitely not normal when you just work by hand. And of course, you can accelerate the target progression from extraction to characterization. So basically, what I'm thinking is that scientists, they should not spend their time, interpreting the data and not labeling all the tubes, balancing all the centrifuge, and preparing dozens of individual conditions. We want to have also some fun in the lab, and we want to be efficient. This is basically what we try to create here. Okay. What you see here is 606. I think today we posted it as a target. We ran this purification at room temperature. So for years we are saying, don't purify at 4 degrees anymore, you can use room temperature. The output is so much better. But for proteins like this, and especially for GPCRs, expression levels are really annoying because they are very low. And you can see on the right side, on the Western blot, okay, we have something, but on SDS page, we don't see it. And what we try to create here is a method where you can actually see on SDS page what you have, because I don't trust Western blots. I want to see what I have, and I see I have nothing. So I was not happy. And we were working on this target for some time. And just last week, my dear colleague Lucas, he upped this thing up to 37 degrees. And I mean, you can see this for yourself. We often see dimer formations in gels. It's normal. At room temperature, you literally only saw dimers. It might be an artifact of the gel. Yeah, I don't want to comment on this. But now you see a wonderful band at the right side of this 40 kilodalton big protein, runs at 43. That's absolutely fine. You can see the band is fuzzy, it's glycosylated, and this looks like day and night. And we didn't do anything on expression level. We were literally just performing the experiment in the Kingfisher with the heating option at 37 degrees. So if today you're working at 4 degrees and maybe you dare to work at room temperature, then maybe it's worth to also go to 37. I have another example, ABCC9. ABC proteins, they're huge and they're very hard to work with. Room temperature, nothing. This was last week, 37 degrees. Yeah. You can see some degradation in some parts or whatever that is. There's an additional band. But for me, this already looks like day and night different. So this is what I want to emphasize to you. And if you would have done this by hand, I think we would have just skipped this target. Like we have so many targets, we have just went with the next one, right, Michael?
Yeah.
But like now, I can just say, okay, it takes an hour, like preparation, let it be 2 hours. We can just do it. Like running the gel is more work than actually performing the whole stream. Okay, now we do see protein bands on the gel. That's already amazing. But what is the protein quality? Is this aggregation? Is it functional? What's the size? We have no idea yet. Okay, so we now pick out those 3 P2X4s, one in SMA, one in ASTI, and one Kubipol. And we have the power triplet here. So we really like the nanotemper devices. With the Prometheus Penta, we can perform a biophysical analysis. And with the Monolith X, we can make binding studies in solution. So we don't have to put anything on the surface. This is how it looks. So if we now evaluate the P2X4 copolymer complex, you can see that it doesn't look alike. So the size is more or less similar. There's maybe 1 nanometer difference. It's also a size you would have been expecting, 8 to 9 nanometer. I know this sounds immensely huge for soluble protein guys, but in copolymers, that's a great size. This is something what we were expecting. And then we did a nano-DSF. So we heat the sample and measure the size at the same time. And we also measure unfolding of the protein by looking at tyrosines and tryptophans. If they expose, when they were hidden before and the protein unfolds and expose, the fluorescence changes. This is something we can then put to the first derivative ratio of 330 to 350, and it gives us this output. And you can see here the stability of the protein over a temperature ramp, which is a direct indicator to the temperature to the stability of the protein over time. Okay, and you see, in glycocobipol, this protein is nearly 15 degrees more stable than an SMA 200, which is definitely worth mentioning because it's much. Okay, so we can now evaluate, okay, the protein has the right size, it looks great on the gel, and it unfolds, so it was folded before, and it looks good. But we don't know if it's really active yet. And so we did a bunch of experiments. Of course, this was done with an upscale of the protein. We now knew, okay, this copolymer looks good, so we upscale it. And we did a blue native gel. You can see this on the bottom left part. So protein should be trimeric. It's like 50 kilodalton in size nearly. It runs around 150 kilodalton, a little bit higher because it's glycosylated. So that's great. So it's trimeric. The DLS size also looks great. Then I said it's glycosylated. So we did a PNG ACE assay to digest the protein. It shifts in size. So, okay, it was also glycosylated. And now comes the master development. Okay, we looked for small molecules. There are no small molecules, for example, base 1797 and 5-BDBD. Those small molecules are very small, between 200 and 300 Dalton. The protein is very big, 150 kilodalton. So this is really something, if you can get the measurement done on a chip, on a surface, that's already amazing. And I want to be frank with you, we worked on this for quite some time. Yeah, because it was not easy. And SPR with membrane proteins is absolutely hard. Also, if you use a GCI, also in this way of rapid technology, this is from Malvamp Analytical, it's not working all the time. There are some problems we have to solve, also the community has to solve. In this case, after we did some optimization, it worked. And it worked very well. So it's expected to be low micromolar, and we can get low micromolar. And we can also use the monolith to do everything in solution. So we label the P2X4, we add the small molecule, and we get beautiful spectral shift data. And this falls on the same range. So we have two orthogonal methods. We know the protein can bind. We know the protein is correctly folded. we know the protein is correctly oligomerized and is glycosylated. So I would say this is one big point for copolymers. It works great. We also tried this out with GLP-1R, very prominent protein at the moment. And we also purified the protein, looks beautifully pure. We checked DLS, looks very uniformous. It's very low in PDI. And then we checked with an antagonist and an agonist, which has low to medium nanomolar measurement with spectral shift. And we also can get this done here. So it doesn't only work on one target after you optimize all the time, it also works on many, many other targets. And if you're a fan of the monolith X and you're a fan of spectral shift, then I hope you can appreciate the nice data we can see here. Okay, so now you know how this works out if you want to screen many, many targets, if you have a Kingfisher. But sometimes this is still too much work. And you maybe want to have everything on one plate. So if I may, I would like now to also introduce plate X. That's okay for you?
I'm fine, yeah.
You're fine with it?
Go ahead, please.
Perfect, perfect. Okay, so we thought, what if you're just starting out with membrane protein work? You have no experience. You don't want to endure all the pain of starting everything from scratch. You don't have an ultra centrifuge. Maybe your lab is not equipped amazingly, but you have a liquid handler. So we were thinking, maybe we can put everything on just 196 well plate. And we have the option here in house, as you saw, as we demonstrated, we can lyophilize the copolymer we produce in house. We can dehydrate magnetic beads, and to lyophilize buffer is not a big challenge. So we created a product where you just have to add your lysate to the plate. You can put it into a liquid handler. We work together with Integra, Assist Plus, and Analytic Jena, the Cell-Biofelix. You just press play and it does everything in one plate for you. So you need even less. You don't have to pipette all the buffers in the plate. You just need a little bit of water, 100 milliliters, and a little bit of lysate, and you're good to go. And this is how it looks. So for this, we really just focus on cubipoles. First, because it's our own invention, and we're very proud of this wonderful class of copolymers. They work out amazingly. We tested them in the lab over and over again. And I told you before, it's the only copolymer backbone which has all these different chemistries around on the backbone, like the glycerol, the sulfur group, a glyco group, a PEG, and an amine. So it can be electroneutral, it can be electronically positive, it can be electronegative overall, like most of copolymers are. And this can have massive benefits, as I'm going to show you in a minute. So we have 8 different cubipoles in one plate. Because different membrane proteins, you saw this, prefer different polymers, but we can still fast parallel process our screen. We have an identical handling across all conditions. It's very high in reproducibility, and everything is on one plate. So you only need one product. There's a little bit more copolymer on there, 45 milligram. There is a little bit more MAC beads on there. Because we can use a little bit more lysate. Here we can use 1.8 milliliter of your lysate, compared to the Kingfisher, where you can only use 1 milliliter. And this is how it looks. It also works for strap, it works for row, it works for flag. You can see if the expression level, like in the LAMP1 twin strap construct, is very good, get a massive output. I quantified it, we have nearly half a mg per mil in the dilution fraction for membrane protein. Yeah? With GLP-1 row, we already saw before how beautifully we can purify it. Of course, in the screen, it doesn't look like it looks after an upscale and polishing, but you can still see it on gel. And we get between 60 to 80 microgram per milliliter in dilution. And we did it with another GPCR, our FlagTech, the GIP-R, and there we get a little bit higher output, around 150 microgram per milliliter. So yes, it also works like this. Of course, all these copolymer constructs, membrane protein constructs, are very uniform in size. They're very low in polydispersity, so the PDI, polydispersity index, gives you an idea of how uniformous the particles in your solution are. And the lower, the better. So something PDI 0.15 is usually something you can get after a size exclusion polishing. This is after elution in one plate, whole purification done by a robotic pipette. Okay. So maybe you're now thinking, it all looks identical. Everything looks good. The cubipoles are giving me the same results. My protein looks the same. Maybe one is a little bit more temperature stable, but I don't care. I just want structure. And we thought so too. We just wanted structure for this. And so we were thinking which copolymer is the right one if everything looks great. So which behaves best on grid. And we saw that we often have issues with preferential orientation, meaning the protein orientates in the air-water interface in just one side. Only get top views, only get side views. But we need a 360 reconstitution, so we need many different
Just got this question in the chat.
orientations. Yes.
Perfect answer.
Oh yeah, okay.
Yeah.
I can foresee it already.
Yeah.
Okay, so what we do if we have a good expression and therefore a high enough illusion quantity, we go directly on the grid. And not like in detergent, we don't need 10 mix per mil anymore. We can get along with half a mix per milliliter. It's also an advantage of copolymers. And we put different cubipoles here on the grid, and you can see a selection of 4 different cubipoles with this P2X4. And you can also see that 3 out of these 4 had strong preferential orientation. The first one, only top views. The second one, only top and bottom views. Also the third one, and now the fourth one. It has a nice distribution over most sides, and you could actually get a structure from one screening condition. Yeah, I repeat, 300 milliliter, 1 purification in a robot on a liquid handling system, also on Kingfisher, looks the same, and you get a structure from this with ATP bound. It got it from the cell, we didn't add it. Yeah, so that's the power of copolymers when everything runs perfectly. This is how it looks. This is what we got from a screening set. It's unbelievable to me. Yes. Okay. So last one to maybe wrap all of this up before we can go into the discussion round. I made a little decision tree because there's many products on our website and sometimes it's a little bit confusing. So what protein type do you want to purify? We're just talking about automation here. It's a soluble protein, amazing. We have the pure HT plates. It's the same technology. It's dried, dehydrated into the plate, but it's less MAC beads. Because for soluble proteins, you don't need as many MAC beads, so you can get away with a little bit of a cheaper price. It's between 2.5 and 5 microliter of MAC beads. We have this for a Histec or for Streptec. Yeah? Okay. But here, I think most of you are working with membrane proteins. Okay, so the next question you have to answer yourself. Maximum flexibility in copolymer screening and selection, yes or no? Yes? Great. This is what we talked about first. This is Kingfisher Flex. Yeah, if you have full flexibility, you order 5 plates, you can pick copolymers how you want them. It's ultra fast. It's 1 hour. You can screen up to 96 different conditions at once. How you want it. Try different buffers, try different copolymers. We made this for Kingfisher. It works like a charm. We do everything on Kingfisher and on the liquid handlers here. If you don't want to think, we did the thinking for you. 16 pre-selected copolymers of our choice only. And you have it for Streptec, Flectec, Rotec. You don't have it for Histec because we feel Histec is maybe not the best tech if you're working in HEC with membrane proteins and you don't stabilize them, so you don't have high expression levels. You will not get great results, so we don't put them in there. But if you're interested and you have a nice result, if you order a few plates, pretty sure we can also do this. Okay, or you say, no, I don't want maximum flexibility. I stick with what you guys say. You are the experts. I want to try out the cubipoles. I have a liquid handler. I have a Sabiofelix in my lab, which we use for other approaches. I have an Integra Assist Plus in my lab. We are using all the time. Amazing. Just check that the components work, and then you can get the Plate XMP. It is the cheapest option for you to get your hands on copolymers, to get your hands on purification. Because this plate alone is cheaper for you than buying the components on their own. I think that's a call for better. Yeah? So it's an all-in-one plate solution. It only needs lysate and water. You don't have to do any thinking. Yeah, it works on liquid handlers. It's 8 pre-selected copolymers for Strep, for Fleck, for Rotec. It's ready. Okay. And I want to sum this all up here because we have success stories. You're going to say, okay, does it work for every membrane protein? I can tell you we have tested everything we can test. It works for every class of membrane proteins. For some it works better, for some it works less. But if you can express it, you can stabilize it and ultimately purify it. Yeah, we have done GPCRs, ion channels, transporters from a lot of organelles, ABC transporters, viral envelope proteins, single pass, multi-pass, only attached via a glycoside, only attached via a lipid, huge complexes, tiny proteins, all of this works. Yeah, and which is particularly to mention, ASMS works like a charm with this. You can do DEL screening campaigns. We have done this. You can do antibody discovery. Yes, we have heard and we know of people, of companies which have amazing results in antibody discovery. And this is like never seen before. You can do lipidomics. If you don't believe us, is there really the lipids inside? Go into PubMed, check it out. There's plenty of papers. It really is. It is really the native lipid environment. And you can also do, of course, small molecule drug discovery campaigns. We've also done this already. Okay, so to sum it really everything up, NativeMP has been successfully applied across structural biology, biophysics, functional characterization, drug discovery, antibody discovery, lipidomics, interaction studies, while preserving this. Yeah, so I would say it's pretty awesome. I don't tell you it works the first time you try it out, but we can help you. We know what we're talking about. We do this every day. We can help you be successful. And we're absolutely willing to help you to be successful. Else we wouldn't be so open with our technology, making it available for everybody. Because we believe that this is really, it's the next evolution in biology. This is what I am absolutely convinced for. I see this every day. Yes. Okay. If you want to have a closer look, we have a preprint. It's on bioRxiv. We have several app notes. You can find all of this on our website. We have worked with MIT. We have worked with Nanotemper. This is really, really nice app notes to also see the power of Monolith, to see the power of Panther, how it works with NativeMP. Please check it out or email us, then we'll help you to find it. And yeah, that's it.
Perfect. Thank you very much, Philip. So we want to have a look
at
the, it's
working.
We want to have a look at the workflow now. So it's still working. I think we will keep this running while we are discussing the next thing. So the poll also clearly showed that most of you, which is not surprisingly, pipe it a lot each week.
Yeah.
Which, as Philip mentioned, we are here to stop or to reduce. And also, thank you very much for your questions. So we have very big engagement. Hopefully we can get through all of this. One thing I want to ask by myself, because I really
we can come back to this later.
Okay, because I really found it fascinating is, so Philip, you did this at room temperature, you saw nothing on your SDS chip. You did it at 37 degrees, you saw it.
Yes.
What do you think is the reason for this?
That's a good question. So yeah, I was puzzled by this myself. But I mean, it's logical, right? Like in the cell, in my body, in your body, everything is at 37 degrees inside of the cells. So this is where the membrane is quite fluidic. If we're talking not about cholesterol, but usual lipids, they are more fluidic at 37 degrees. And of course, it's thermodynamics, right?
So, yeah.
It's thermodynamics. The copolymer is amphiphadic. It has negative and positive charges. And the membrane inside is hydrophobic and outside is hydrophilic. So it's also charge dependent. The copolymer has to find a way inside of the membrane. So we have to introduce energy to make this process faster. Yes, you saw that we reduce drastically the solubilization time to make it more convenient and because it works. Yes, so if we adapt or make the temperature higher, we put more energy inside of the system, so it makes it more favorable for the copolymer to stabilize.
Yeah.
And if we make sure that the protein is stable in its environment and doesn't degrade within 5 minutes, like it sometimes does in detergent, I don't know if you've ever purified a membrane protein in detergent at room temperature, it's a bad idea, I can tell you so much. Because maybe it works in 1 out of 50 cases, but the majority you just get rubbish out. So yeah, I would explain it like this. And of course, you have to be careful that the protein doesn't affect its folding, but for this you then have to make assays, make a binding assay if it still binds. But if you have a chance to purify a protein nobody has ever purified before, and you have to use higher temperatures, which it has been it has been living in since it was overexpressed. It makes sense, right?
Makes totally sense, yeah. And for me, I mean, this is because some customer also told me this, like the robot is kind of magic, and you would also agree this would not be possible doing it by hand, right?
I can tell you it's not possible, not in this time frame. It is also like the mixing of the robot. Is it a pipette or is it this Kingfisher arm which goes up and down very quickly? It mixes the solution so quickly that we have a feeling that you get a better exposure of the MAC piece to the solution. You get way better binding. It's way quicker, also by introducing energy.
Yeah, no, it's really fascinating and not fully understood yet. So yeah, also please feel free to do research on that. So we now go ahead and answer the question from the chat. The first question goes to Bernard. So he asks, my company's next project involves a membrane protein complex, not a singular protein. Will NativeMP give me the whole complex or will it tear it apart? Beautiful question. Thank you.
Yeah. It's a good, it's an important question.
Yeah.
Okay, let me answer like this. There is a very recent preprint from Matthias Mann Group. He's the one and only god of mass spec. Yeah,
he is.
And we've tried to influence them to use our copolymers for a very long time. And we have a product that's called Gentleize. It is a lysate buffer, which is 100% detergent-free on copolymer basis. So it's basically like copolymers, but convenient in a buffer. And they've tested this out. They have tested different detergent buffers against our Gentleize. And they use TNF alpha, which binds to TNF receptor, to pull out TNF receptor and maybe intracellular components. And with detergents, they could sometimes catch TNF receptor, but that was it. But with Gentleize, they were able to catch TNF receptor and intracellular interaction partners. So that's a first strong hint that it's pretty gentle, like the name suggests. We can see that we get heterodimers purified with NativeMP, which do not work out with detergents.
What was the biggest proteins you got so far?
The biggest proteins we got out, well, yeah, I think this is H-ERG is huge. It's like a tetramer, if I'm not wrong. Okay. And it's like half a megadalton nearly. But even more impressive is, for example, the amylin receptor, calcant RAM3. So it's a GPCR heterodimer, and this is very delicate, and it works like a charm with NativeMP. So I would say the chances are very high, and you don't have to be so gentle like you think. Like sometimes with sonicating samples, like every time we open cells, it doesn't really harm it. Go up with the temperature, just like beat it a little bit, and just see if it works out. There are many examples where great complexes have been successfully purified in copolymers. And also a nice thing is the size of the disc is adaptable to the hydrophobic core of the membrane protein complex. So it's not like with MSPs where you worry, is it too big? Is it too small? Yeah. It's just the right size.
Yeah. And also what I want to add here is like, you also have to screen for it. There are different polymers that makes different. So that's also very important here. And what we see is that especially with big complexes, sometimes they are surprising copolymers that don't work very often work.
Yeah, absolutely. Yeah, sometimes sulfo ones, which are not the best in solubilizing, work out and stabilizing here. It's not a magic bullet, right? You're not going to do it. Yeah. Hopefully it works first try, but like it's not a magic bullet. But if you put some energy in it, you will be successful with it. I'm pretty sure of that.
Let's go to the next one. So we try to answer every question. Let's go. From Xavier, can the liquids in the screening plates can be dispensed using a non-contact dispense system such as Revity FlexDrop or an Echo? Or does it have to be pipetting robot similar to the Kingfisher with a 96 tips head?
Okay, so what we've adapted it on is those two systems. They use an 8-channel pipette and they pipette all the solution. You have to use something which mixes the magnetic beads with the solubilizate, and first of all, which mixes the lyophilized copolymer with your solution. And therefore, you need a pipetting step, at least as far as I know at the moment. So there's no option yet to have a non-contact dispense system. Maybe in future, this would be an option. I am curious what the benefits would be in your eyes, compared to just pipette it.
Yeah, but I can tease that we also are in contact with Gravity to work with their system, but that's all I can say for now. Yeah, but as Philips, I also totally agree what Philip said. The next question is from Abha. Hi, I think I missed it in between. With this procedure, are you getting rid of native lipids from the membrane? How copolymers eventually solubilizing the membrane protein? Yeah.
Okay. So we're not getting rid of native lipids from the membrane. The copolymers, they randomly introduce themselves into the membrane and they randomly cut out nano discs. So they cut out membrane patches. By chance, you're going to have your membrane protein in there, and then you use the affinity tag to pull on it and purify it. And what we see is, even though it's described that this copolymer nanodiscs in the very beginning are heterogeneous in size, once we purify them via the affinity, you saw it, very monodispersed. So they're very uniformous in size after purification, but it's a random process. So it's not targeted to a GLP-1R. No, the copolymer doesn't know what it does. It just like solubilizes everything. Solubilizes every membrane it comes across. Just thermodynamics.
Then another question from Bernard, and that's also a question that I asked Philip sometimes by myself, because customer also ask very often with, my construct carries the HISTEC. Are you also offering the plates with HISTEC beads?
Yes, yeah. Not at the moment. As I said, we can do custom plates for you with HISTEC. There's no worry. We have this beautiful indigo resin, which works a charm with soluble proteins. And if you have very good expression level of your membrane protein. So we did some tests with HISTEC. If the expression level is beautiful, it works here. But the thing is, most often it is not. And we don't want to overpromise something. So if we show you these examples where we used antibody tags to purify super hard to express targets, and it works out, and I sell you a plate with a HISTEC, and you do it, and it doesn't work out, you're going to ask me why. And if I then tell you, okay, it's because of the HISTEC, you're going to be unhappy. So I'm going to be upfront with you. If you have a good expression level, we can get something going. I'm pretty sure you order a few plates, and maybe you're going to be successful. But if you're often working with very low expression, I can only advise you to use Streptec, Protec, Flectec. I know the resin is more expensive, but the question is, what is more expensive? Getting the expression properly done, doing all these modifications to your protein, or using another resin in the end.
Also wanted to emphasize, so I mean, it's clear we also producing HISTEC by ourselves, and we don't produce Flectec by ourselves.
No, we don't.
But we're still offering Flectec here, not HISTEC. So that really should also give you the hint. It's really very often not working well with HISTEC. Yeah, even if we really want to have it.
Maybe this is also different for bacterial proteins, but here in house we're usually working with eukaryotic cells, and
we have two similar questions from Suparat and Ruchika, and it's about the yield. So what is the range of final yield at the end after the elution step? I mean, it would be good, I think, if you could answer it like, how's it normally per well? And then you could sum it up to 90.
Okay. Yeah. So elution quantity is 50 microliter per well. Depends highly on expression level. Yeah, I showed you before. It can be everything between nothing and a mg per mil.
And a mg per mil. Per well.
Yeah, a mg per mil per well. But it's only 50 microliters.
Yeah, but that's crazy.
Yeah. So what we had with P2X4 was, I think, about 450 microgram per milliliter. We had 50 microliter aliquots, and we used this. We just did desalting, nothing else to the sample, put it on the grid, worked out.
Okay.
Yeah. But with GPCR, sometimes it's only 40, 50, 60 microgram per milliliter. But then still you're going to use biophysics. You're going to see it on the SDS page, Comasi, whatever you want to do, and you get an idea what is the right thing, and then you can upscale it.
Okay, we have not much time left. So Philip, the next one we have to answer a little bit faster because I want to have them all. So this one you answered from Lucas, the orientation bias, different polymer nanodiscs doing cryo-EM. Do you have any suggestions? So as you see, we see it, and the suggestions are, Philip?
Sorry again. Yes. The suggestion is try different copolymers. So do the screen, put them on there, we have an idea that the cubipoles might work nicer in many ways. So especially the glycerol and the glycocubipole.
Polymers that tend to be less charged, right?
Polymers which tend to be less charged, yes.
Good. Next question is from Xavier. Just to be clear, the NativeMP plates are single-use or can be reused if the buffers are refreshed?
Okay, the NativeMP plates are designed to be single-use because the copolymer is going to be gone after you used it, but you could recycle the magnetic beads, of course, and reuse them. Yeah, and if you buy the copolymers and you put the stock inside of there and you recycle the mag beads and you put all the buffers in there, yeah, sure, you can reuse it. Yeah, I just want to quickly emphasize we are now exactly at the moment where the elution happens. So it's a GFP tag protein, and I hope you can see that it's nicely in green, and the Kingfisher is just collecting the mag beads from there. So what is happening right now is the end of the purification, while we're coming also soon to the end of the webinar, and you see that it worked beautifully. And you can do this with 96 different words. You can see there, mag beads are getting out. It's nice and green. So purification worked out a charm. And now we can go back to questions.
The next question is also from Xavier. Why can you only use less than a mg of protein in nanodisc compared to detergent where you need way higher concentrations?
That's a good question. And I honestly have no idea. Maybe it's a different orientation in air-water interface. If you have preferential orientation, a good idea, at least our cryo says this, is that you can add a little bit of OG to the sample, and then again you need way higher concentration. So the detergent does something, but sorry, I'm not an expert in cryo, so I don't want to tell you any wrong information. I just can tell you that we have shown over and over again that it's definitely the case. You need less.
Yeah, but perhaps you can also connect you to the cryo.
Yeah, of course.
Expert there. Then next message from Lucas. Do you have suggestions for native nanodisc membrane proteins which have no cytosolic domain? Is there anything you found helpful for 2D class alignment? That's a very good question. Because, for example, for a dimeric protein, the side view will be fine, but the front view will only feature a relatively small protein part, but a large nanodisc part, yeah.
You're right. So yeah, you need some kind of an anchor. You need something which is bigger than 30, maybe 40, 50 kilodalton, which anchors the protein from the outside so you can get proper 2D classifications and then later get the structure done. For GPCRs, people are often using G proteins, or there's other options to enlarge the ectodomain of the protein. But if you only have a domain which is within the membrane, you're going to have a very, very hard time to get the structure out.
Then I think also this is what we discussed also today, and we'll discuss, I think, every day, because Philip is working on it every day. What challenges you still face with the copolymers? Can you point towards some specific limitations?
Yes, yeah, of course. It's not our magic. It's not a magic bullet, as I said. It's I think I'm convinced it's the future, but it's not a magic bullet. So everything in solution works amazingly. Spectral shift works great. ASMS, affinity selection mass spectrometry. Our collaboration partner got results you wouldn't believe it, and we're soon gonna showcase them. This is crazy how good this works. Del ability, Del screening works like a charm. Everything which is on a surface is a challenge. I'm going to tell you this honestly. It's a challenge in-house, it's a challenge with our collaboration partners, and it's a challenge in pharma companies and in academia. If you have to put it on the surface, you have a complex from a protein, you have lipids, you have a copolymer. Everything is somehow charged, everything is somehow hydrophobic. And if you then want to add small molecules or peptides, which are very strongly charged, you're going to have a very hard time with unspecific binding. So we are working with all forces we have to solve the issues in this case, in this regard. But this is definitely a limitation. But it's also a limitation with membrane proteins and detergent, because the detergent micelle also is hydrophilic and hydrophobic at the same time.
Yeah, and I think I have a last question from Eduardo. The other ones will be answered via email, don't worry. So no question will be not answered. But where is the question again? Here, there was again another question. And the
it's not up yet.
It is not displayed. How did you confirm the presence of RAM3 along with the CTR?
Very easy. It has a tag, and we purified via one tag and pulled the other protein with it. And then we did a big campaign to find binders, which only bind if the protein is in the dimeric, heterodimeric conformation, so we can be sure that the RAM3 is there. It's way smaller than calc, so yeah. If it's there, you can see it on the gel. You saw it on the gel. The lower band was RAM3.
Okay, good. Then with this question, we close it. Thank you very much that you were so engaging that we even had 20 minutes of question here. So as I mentioned, we will answer them everyone afterwards with the experts and also connect you with the cryo-EM experts. If you have any question, reach out to us. You can also find us on LinkedIn, connect with us there and discuss with us. We are really here also to move the field forward. Nothing is like easy and works on the first try. And yeah, let's push it forward together and hopefully see you in the next webinar on LinkedIn or on the conferences. Yeah. Wish you a nice evening or good day wherever you are. Yeah. Bye-bye.
Bye-bye.